Photosynthesis is the process by which plants harness light energy to synthesize organic compounds. It is made up of two steps, light reactions and Calvin cycle. Before we go into the steps, here is the chemical formula. Notice how carbon dioxide and water react with sunlight to form glucose and oxygen. CO2 enters and O2 leaves through the stomata, which are tiny pores on the underside of the leaf. Photosynthesis is performed by autotrophs, which is basically a term for self-feeders. Autotrophs are plants, cyanobacteria, and algae. This reaction takes place in the chloroplast of the plant cell. Notice the stacks of green discs. These are the thylakoid.
So now on to the two stages of photosynthesis. The first step is light reactions. First, we have a photon of light energy that is absorbed into the thylakoid. This energy from the photon enables an electron to be transported through the electron transport chain to create ATP and NADH. We will go over the electron transport chain later. Once this occurs, a water molecule is broken apart, and then the free hydrogen ions are used to make ATP and NADH with oxygen as a waste product. Secondly, we have the Calvin cycle. This step is important because it makes glucose from the products of light reactions. It takes place in the stoma of the chloroplast and converts CO2 into sugar. It begins with the input of CO2. In this cycle, the products from the light reactions reduce 3 molecules of CO2 and produce 1 molecule of G3P. These G3P molecules are the final product and combine with each other to make glucose. The Calvin cycle must occur twice for glucose to be created.
Plants not only perform photosynthesis, but they perform cellular respiration, just like us humans. Cellular respiration is the process by which cells break down the food they take in into ATP. It takes place in 3 phases: glycolysis, the Krebs cycle, and oxidative phosphorylation. The first step is glycolysis. This step is when a glucose molecule is split into 2 pyruvic acids, which are used in the next step. It also produces NADH, which is an electron carrier, and ATP.
Next, we have the Krebs cycle. The Krebs cycle takes the 2 pyruvic acids, modifies them, and then breaks them down to create ATP, NADH, FADH2, and CO2. Notice that for every glucose molecule, there are 2 pyruvate molecules that enter the Krebs cycle. NADH and FADH2 will be used in the electron transport chain. So this leads us to the last step in cellular respiration, which is oxidative phosphorylation. This includes the electron transport chain and chemiosmosis and takes place in the inner membrane of the mitochondria.
NADH and FADH2 are electron carriers and are oxidized as they give their electrons to the protein complexes in the electron transport chain. The electrons are then passed down the chain through redox reactions and are eventually received by an oxygen molecule. Because the energy given off by the redox reactions can be used to pump H ions out of the membrane, there is a higher concentration of H ions on the outside of the membrane. This creates a concentration gradient. Chemiosmosis is when, due to the concentration gradient, ATP synthase harnesses the energy by allowing the H ions to flow down the gradient. You can think of this as similar to a dam. Through ATP synthase, the energy to attach P to ADP and form ATP is created.
So what are the connections between photosynthesis and cellular respiration? Photosynthesis and cellular respiration work together because the reactants of one are the products of the other, and vice versa. Photosynthesis's products are glucose and oxygen, and cellular respiration's products are carbon dioxide and water. Photosynthesis uses carbon dioxide and water created by cellular respiration to create glucose and oxygen in cellular respiration. And cellular respiration uses the glucose and oxygen to form carbon dioxide and water. Humans eat the glucose made by the plants and then through cellular respiration convert it into energy they need to survive.
Now that we know about the electron transport chain, we can look at something that could go wrong in this process. As we explained earlier, the concentration gradient is essential for the oxidative phosphorylation to create energy through ATP synthase. So what happens when this gets messed up? In the 1930s, DNP was considered an effective diet pill. But now that we know more, we know that this is a thermogenic decoupling agent and can have serious harmful effects on your body. While it does increase your metabolic rate, the uncoupling agent causes there to be no concentration gradient in the electron transport chain. How does it do this?
A thermogenic decoupling agent disrupts the electron transport chain and stops the body from producing ATP because the concentration gradient is now uncoupled and does not exist. Protons do not go through ATP synthase, and most of the energy produced through cellular respiration is lost as heat. DNP is an inhibitor of ATP production in the mitochondria cells. While we normally depend on the concentration gradient created by the pumping of H+ ions across the membrane using the energy from the electron transport chain, DNP acts as a proton 4, allowing the protons to leak across the inner mitochondrial membrane and therefore bypass the ATP synthase. So why is this bad?
Our bodies do not create enough energy. Because DNP prohibits oxidative phosphorylation from occurring, large amounts of energy is being consumed without a sufficient amount of ATP being produced. For your body to get enough energy, your body has to increase its metabolic rate in an effort to create enough energy. Therefore, more fat is burned to compensate for the inefficiency of the electron transport chain and to meet energy demands of your body. That is why this is used as a weight loss supplement.
So yes, this is an effective diet pill, but we now know how critical our electron transport chains are. So it would be best to stay away from any decoupling agents. DNP might get rid of fat, but it can also kill you.